Howdy, folks. No, this background shot here is not of my school closet or any of my workspaces. I'm painfully disorganized in that regard. Heck, I can show you in the background here. I make sure that I have the organized part of the clean part of my office in the background. You don't have to go too far to see that. Maybe I could, use some help in the organization department. Anyway, sorry about that. Hope I didn't get you sick. Um, Thanks for checking into Mr. Ulrich's Land of Biology. I am Mr. Ulrich, and no, we're not going to be talking about office organization in this video. We're going to be talking about genetic organization, how material DNA is organized inside of all of the nuclei of all of the cells of your body. Most of my students come to me already knowing that DNA stands for deoxyribonucleic acid, and it's the genetic material that is found in the nucleus of our cells. When I ask them more specifically what it's for, I start to get answers like, it's what I'm made of. And you're not actually made of DNA. There's DNA in you, but it's not really a structural component of you. It's probably more accurate to say that you're made of proteins. Yeah, you're mostly water, but so is Kool-Aid, and so that doesn't tell us very much. Sometimes students will tell me that it has certain traits encoded in it, like it says that I have blue eyes, and I have blue eyes. But I have blue eyes not because I have a gene for blue eyes. It's actually because my gene for brown eyes is broken. So it's a little more nuanced than that. Now, when students say that it has information about how to build different parts of my body, you're starting to get closer. And when we start talking about proteins, that's really what it is. DNA is the instructions for protein construction. And it's really important that we get this connection between DNA and proteins. Well, if DNA has those instructions for protein construction encoded in it, how exactly does a code like that work? Well, it works the same way that we encode our language using symbols. In the case of English, we use a Latin alphabet, but it's not the only alphabet. The Korean alphabet, the Cyrillic alphabet, which is used by Russian languages. Sure, all of that works. Heck, you don't even have to have like an actual alphabet. It can be random weird code like this. All you have to do is just be able to translate, is to convert from one sequence of characters to another sequence of characters. The characters are really largely arbitrary, and so that we can translate then from one sequence to a different sequence and just say that it has the same meaning. Where we encode information by putting a sequence of characters on a piece of paper or a clay tablet or a computer screen, clearly that's not how DNA works. DNA is a chemical, and so it's going to have its code as a form of a sequence of chemicals. In this case, those chemicals are called nucleotides. So DNA is actually a string of nucleotides. We call that a polymer because the poly for many, lots of nucleotides in the row. It's actually called a polynucleotide. Each nucleotide is then the monomer of the polymer that is DNA. So each nucleotide, each monomer has three parts. It has a sugar, that's the deoxyribose. Attached to the sugar is going to be a phosphate group. And then also attached to the sugar on the other side is going to be what we call a nitrogenous base. And there are, there's four here in DNA. There's actually a fifth one that we'll come into when we get to RNA, but that's later on. All right, and they're adenine, guanine, cytosine, and thymine. And we don't really need to know their full names. You just need to know the first letters, A's, G's, C's, or T's. Now, what this means is that a lot of each monomer is absolutely the same as the next one. The only difference then is the nitrogenous base. And just like in our letters, there are certainly characteristics that are all the same amongst these letters. They are all kind of flat on one end. They're all lined up. They all are black in color. They don't have serifs, right? There's all kinds of things that we can say about characteristics about these things that are the same. But there are also things that are different. There's enough different that we can recognize the pattern. And there's enough difference amongst different nucleotides with the nitrogenous base that we get a pattern of A's, G's, C's, and T's. So when we start talking about the code of DNA, we forget about the phosphate and the sugar because those are going to be there in all of it. That's like the paper that it's written on. And we really focus on the order of those nitrogenous bases. So we can kind of think of the language of DNA as only having an alphabet of 4, and if we count uracil and RNA, like I said, we'll talk about that later on, of 4 different letters. Now, there's more to the structure of DNA that we need to be able to recognize than just being a sequence of bases. It needs to be stable. Stable enough to not fall apart, but unstable enough to be able to be unzipped by enzymes. I'm getting ahead of myself. We'll get to that later on. Most of my students do recognize that DNA forms this double helix or twisted ladder structure. What we need to start to be able to talk about is the how those monomers of nucleotides line up to form the ladder that's going to twist and form this double helix. The legs of the ladder are those sugars and phosphates, the same parts. The rungs of the ladder, the inside, are where we're going to find the nitrogenous bases. And now you might be saying to yourself, hey, but we got nitrogenous base and then I got sugar phosphate on one side. How do I get the sugar phosphate on the other side? Well, DNA is not just a single strand of nucleotides. It's actually a double polymer. So I got one strand on one side, and then I have another strand of monomer of nucleotide on the other side. And then they have their bases in pairs in the middle. So here's a G and a C, and here's a T and an A. Now, the reason why I have these dotted lines here between the G's and C's and T's and A's is because these bonds are different from these bonds. Now, again, this is where it's really helpful to have talked about chemistry in class before we start getting into DNA chemistry, but it is what it is. Hopefully this will help you understand it when we get to it in regular bio a little bit better. So these covalent bonds over here that are the solid lines, these are much stronger and more stable, and they tend to not fall apart. These are what we call hydrogen bonds in the middle, and they're quite a bit weaker, and they're a little bit easier to take apart. And that's a big part of the functionality of DNA, is its ability to come apart right down here in the middle, right at the bases, and come back together again as well. Back before Watson and Crick figured out the structure of DNA, Chargaff was also looking into the structure of DNA and what it's made of. We knew that DNA, he knew that DNA was made of nucleotides, but how the nucleotides and the sugars and the phosphates were all arranged was not clearly understood. What he did is he just took DNA from all different types of organisms and broke it apart into its little pieces and measured up the amount of adenine, thymine, guanine, and cytosine that was there. And it didn't matter what he took it from, he always had the same ratio, the same amounts of adenine and thymine, equal amounts, as he had guanine and cytosine. Didn't matter what organism. And so this led to Chargaff's rule that T's always bond with A's, thymine and adenine always bond together, and guanine and cytosine always bond together. There's only room in between the two strands for 3 of these little rings. And so I'm always going to have a 1-ring structure and a 2-ring structure. There's called purines and pyrimidines. You don't need to know that. The hydrogen bonds in between also keep them apart. Adenine only makes 2 hydrogen bonds, where cytosine wants and guanine here make 3 in between them. And so that's how we keep the fidelity, maintain the fidelity of the code, even as it unzips and is read and goes back together again, or copies are made of it. So just like it's the sequence of letters and spaces that makes the difference between Shakespeare and Kanye, it's the sequence of A's, T's, G's, and C's, nucleotides, that's going to be the difference between me and my dog, Roger. I just mentioned Watson and Crick in passing before. They're the two researchers who were given credit for determining the structure of DNA, the whole double helix with the bases on the inside. They, of course, couldn't have done their work without Rosalind Franklin's work, but that's a whole other conversation. We can have that. They immediately recognized with the structure and Chargaff's rule that there's this semi-conservative model of making copies of DNA. We call it replication. That as long as you unzipped the DNA in the middle, the bases would stay in sequence, and all you would have to do is move in new nucleotides, complementary nucleotides, and you'd end up with two double strands of DNA that, well, the two new daughter strands each will have one DNA side chain that's old and one that's new. And that's why we don't call it duplication, we call it replication. And it's semi-conservative because I don't end up with a master and a copy when I'm done with it. It's very hard for our little feeble, limited chemical reactive brains to really comprehend the scope of information that is conveyed and carried in DNA. The amount of information necessary to make all of the proteins for any organism, not just a big human or a big elephant, but an amoeba, is amazing. We have upwards of 3 billion base pairs in each one of our cells, nuclei. That's 5 feet of DNA in each nucleus. And it has to be not just crammed in there like y'all do with your notes and your book bags, right? It has to be carefully put in there so that each thing can be taken out, each part of the DNA can be unzipped and read, and it has to be read by stupid cell parts. They're just enzymes. They can't think. They can't go, wow, this needs to be done differently, right? They just do, right? And so how do we cram all this information into the nucleus of the cell and keep it somewhat organized? Well, most of you guys have heard of chromosomes. You've seen pictures of chromosomes, the little X things. I'm here to tell you they're not always X's. This is what they look like right before cell division. All right, so we have these chromosomes. What they are, the chromosome is the result of the condensation, the coming together of all of that DNA. We call that loose DNA before it's a chromosome. We call that chromatin, right? And the chromatin condenses together and forms chromosomes. Now, these X's, like I said, are right before cell division. And so there's 2 legs. Each leg is called a chromatid. I know. Chromatin condenses into chromatids. 2 chromatids makes a double chromosome. Lots of chromas makes some flashcards. All right. Now the sister chromatids, like I said, because we're talking about right before cell division, so replication has already taken place. There's already a copy for each daughter cell. Right? The chromatids are the result of that replication. You start out with 1 piece of DNA, it unzips and becomes 2 pieces of DNA. It actually stays attached right here in the middle. It's a structure called the centromere, stays right there. But that means that the 2 sister chromatids, because they're the results of replication, have exactly the same base sequence on it. They carry the same information. Now, what we can do is you can theoretically grab onto the end of one of those chromatids, right, and unravel it. Right, just like on the cartoons when the mouse grabs onto the hair on the tail of the cat, ends up with a big pile of cat hair and the naked cat, meow, right? We could kind of do that here with a chromatid, only you're not going to end up with a naked cat. And take a look at the way that all of the DNA is shortened to fit in there. First off, we take the ladder and it's twisted into that double helix. Then the double helix is then wrapped around these histone proteins to make they act like spools, and so you get like this string of pearls. Right, here's one here, here's one here, here's one here, here's one here, right? And then you take that string of pearls and coil that up into a foam cord. And you take that phone cord and you loop it back and forth in these loop domains, and there's all these different ways that it crams together just to shorten all that information and get all of that base pair sequence in there. All right, well, that's enough for now. We'll spend some time in class talking about replication and the process of replication, and I'll try to walk you through that so it makes a little bit more sense, and then we'll use some of these terms more, and you'll get used to using them. But we'll stop there for today. If you have any questions, you can hold them for class tomorrow, or drop them in my email, or put them right in Google Classroom, whatever. Other than that, I'll see you in class.